Hello, my name is Antonio Baeza. I'm an associate professor at the Department of Biological Sciences, Clemson University. And in this talk, I want to uh, give you some information about testing the utility of low-pass nanopore long-read sequencing for mitophylogenomics and barcoding research using the Caribbean spiny lobster Panulirus argus as a model system. I want to start by mentioning something that is no news to you really. The mitochondrial DNA, the entire chromosome or small fragments of it represent or are a marker of choice during the last 20 years to understand population genetics, phylogeography, connectivity, demographic history, among many others, in marine and terrestrial vertebrates and invertebrates. Um, what I want to focus during this talk is about the methodology and how to improve the retrieval of entire mitochondrial genomes profiting from third generation sequencing. So I was mentioning that short fragments or the entire mitochondrial genome is a marker of choice. Has been a marker of choice during the last 20 years. And usually what you do to retrieve information from these markers is PCR and Sanger sequencing. If you're interested in retrieving or sequencing one or a few fragments, everything is straightforward. Bench work is not too heavy, but turns out to be time consuming and heavy if you're interested in sequencing the entire mitochondrial genome of an invertebrate or a vertebrate species. That's why it's important, or that's where we can profit nowadays from second generation sequencing technology, short read sequencing from Illumina, for instance, the start or initial read sequencing error has improved so much nowadays that using a low pass or a low coverage strategy, we can sequence and assembly entire mitochondrial genomes, spending not a considerable amount of money. The problem with Illumina is that it's very time consuming. You can start a project or from initial sampling of a specimen or um, purification of DNA all the way to that analysis. That project might take several months or sometimes half a year. It's very important for me for answering many applied questions in conservation genomics to be able to retrieve markers very quickly. Excuse me. Unfortunately, Illumina does not allow us to do that. And that's the reason why several colleagues during recent years, during the last 5 or maybe 3 years, are increasingly relying in third generation sequencing technology to be able to sequence complete mitochondrial genomes. Nowadays, we have long read third generation sequencing technologies such as the Oxford Nanopore Minion or devices developed by Pacific Biosciences that indeed allow us to produce a relatively large amount of data. Actually, not relatively, is indeed in absolute terms an impressive amount of data in relatively short periods of time. The problem with third generation sequencing technology is that still the initial sequence error rate is too high, considerably higher than that error reported for Illumina machines. The other problem, especially with the PacBio, is that The device itself is extremely expensive. You need very high quality DNA for library preparation. It turns out expensive to retrieve that high quality DNA from samples on the one hand. Library preparation is also time consuming. And again, as I was mentioning, this technology is very expensive. On the other hand, and on the other extreme of this third generation sequencing technologies devices, we have the Oxford Nanopore devices, especially the Minion. The device itself is cheap, relatively speaking, and very cheap when compared to the machines developed by PacBio. Nowadays, we don't need extreme high quality DNA to prepare the libraries. The protocols have been well developed for the extraction of DNA or RNA, if you're interested in sequencing RNA in these devices. And we have the possibility of, again, sequencing in a device for 24 hours or 48 hours. So in a relatively short period of time, have a large amount of information, and then use that information for assembling mitochondrial genomes. So I see the Minion as disruptive technology. It has the ability, for instance, to provide access to sequencing technology to countries, low income or moderate income countries, so that colleagues in these countries in Latin America, for instance, can have access to these tools, so to answer important applied conservation genomics locally, not relying on developed countries on the one hand. So what I want to do in this talk is, I have three main goals. The first one is I want to test if mitochondrial genomes can be sequenced using low coverage, long read nanopore sequencing data exclusively. I want to highlight here that I'm very interested in the low coverage idea here. We are trying to optimize resources. We're trying to minimize the cost for sequencing mitochondrial genomes. So we are interested in testing if we can retrieve entire accurate mitochondrial genomes using low coverage data, low pass sequencing, whole genome sequencing. And also I want to highlight the idea of exclusively. There are several studies, and they are increasing every month, using long reads, Nanopore and PacBio, together with short reads for the assembly of large genomes, including the human genomes, bacterial genomes, viral chromosomes. However, to the best of my knowledge, for mitochondrial genomes, there's no study so far, especially in the arthropoda, a really very diverse group of invertebrates that has used long reads exclusively to retrieve mitochondrial genomes. The second goal is to explore the accuracy of these long read assembled mitochondrial genomes. And I will do that here by benchmarking the long read assemblies versus what we call a gold standard reference mitochondrial genome obtained via using Illumina short reads from the same individual using the same DNA for the assembly of long read mitochondrial genomes. And last but not least, I want to test if these long read assemblies, mitochondrial genomes, irrespective of the accuracy, that might be high or low, we will see, are useful for mitophylogenomics and for barcoding research. Now, to accomplish these goals, I will be using the Caribbean spiny lobster, Panulirus argus. Let me give you a little of information about this species. These large, clawless, or spiny lobsters is the most economically profitable fishery in the Caribbean. The industry is approximately a $1 billion industry. So you can imagine that there's a lot of local communities and people relying on this resource all over the Caribbean. on the one hand. Um, Unfortunately, all the populations in this species are either fully exploited or overexploited, and there are serious issues of mislabeling at multiple steps in the supply chain of this resource. So if we are able to tackle that problem, we should be able to improve the management of this fishery towards the goal of sustainability, and also to try to understand the effect of overexploitation on these different populations. So as you can see, there's many reasons to generate genomic resources for these very important species, marine species in the Caribbean. And of course, I want to highlight that this is a species very important from an ecological point of view. This species is a prey of many predators in coral reef systems, in hard bottom environments, shallow environments, in tropical or subtropical areas in the Caribbean, including the Florida Keys, animals all the way from sharks among vertebrates to octopi among invertebrates prey on small lobsters or on individuals of moderate body size on the one hand. And this species also attain a relatively large amount of energy from leucinid clams that rely on chemosynthetic activity, actually. The point here is that this lobster is transferring energy between different communities uh, in shallow environments, between seagrass meadows where leucinid clams live and coral reefs, but also is diverting energy from chemotrophic environments to other communities that mostly rely on photosynthetic activity. So again, from an ecological point of view, generating genomic resources is very important in this species too. And here we're interested in, we're focusing on mitochondrial genomes. So let me tell you first about testing if complete mitochondrial genomes can be sequenced from low coverage, long-read nanopore sequencing data exclusively. And I will speak also about the accuracy of these mitochondrial genomes if we're able to retrieve them then. So the first thing that we did in my lab was to generate a gold standard. We sequence using well-established protocols the mitochondrial genome of this species using short reads. We extracted DNA and using Illumina technology, specifically almost 500 million reads of parent short reads, Illumina short reads. We were able to assemble the mitochondrial genome of this species. The tools that we used were several, including MitoBeam, including Novoplasty, and including Get Organelle. Novoplasty and Organelle are bioinformatic tools specifically developed to assemble mitochondrial genomes from short-read datasets. The amount of data of reads that we generated allow us to assemble a high-quality mitochondrial genome with a coverage between 710 and 830x, depending on the pipeline that we use. We annotated this circular mitochondrial genome assembled by Novoplasty and by Get Organelle with Mitos and Mitos2. And the annotation shows us what we were expecting. In these mitochondrial genomes, we have 13 protein coding genes, 2 ribosomal RNA genes, 22 transfer RNAs, and also a relatively long region that is the putative control region. And this is what you expect for any arthropod. This is what you expect for any crustacean and any spiny lobster in particular. And the point here is that we are able to, as expected, assemble a high quality gold standard mitochondrial genome using short reads. If we compare gene synteny in this mitochondrial genome, we discover that it's exactly the same reported before for all the other species for which other colleagues have assembled mitochondrial genomes. Another reason, again, to indicating that the quality of this mitochondrial genome is high and that we can use it as a gold standard so to compare the quality or the accuracy of long-read mitochondrial genomes. So let me tell you now about the different strategies that we use in an attempt to assemble a mitochondrial genomes using low coverage nanopore long reads. Using standard protocols, well established, we extract the DNA, we prepare libraries, we barcode them, and we sequence the library that we were able to produce for the Caribbean spiny lobster, same individual from which we obtain short reads. We sequence this library in a single minion for a total of 48 hours with 5 other libraries. And again, the idea was here to produce in a relatively short period of time a data set that we consider is a low pass or a low coverage data set. And the question then is, can we, using this data set, can we generate relatively quickly a mitochondrial genome for the same species, for the same individual? So let me tell you a little about the pipeline. First, we were able to obtain a total of 77,520 reads after running the sequencing for 48 hours. Base calling was conducted with albacore. For trimming, we use pork chop, and for quality control and filtering of these reads, we use FastP. Before telling you about the different assemblers, the different assembly tools that we use to attempt to retrieve the mitochondrial genome of this species, I want to mention that we use albacore. The base calling happened during the year 2018, and at that time, the version of Guppy wasn't performing better than albacore. And because of that, we just decided to go to albacore, that at that point in time was the established base caller, or one of the most efficient base callers that we did have access to. So about the assembly of this, the assembly strategy, we use 3 different tools: Fly and Unicycler for de novo assembly, and Reballer for reference-based assembly. In Fly, we polish the assembly with the Fly Polisher tool, part of the Fly pipeline, 1, 5, or 10 cycles, for 1, 5, or 10 cycles. The idea here was testing the effect of the number of polishing iterations on the quality, on the accuracy of the assembled molecule, a mitochondrial chromosome. For Unicycler, we use 3 different strategies: bold, normal, and conservative. They are part of the Unicycler pipeline. And for Reballer, we use 3 different sequences or species as reference. We use the short read assembly that we just generated for the same species as a reference. And also we use Panulirus cygnus and ornatus as other 2 reference species. Panulirus ornatus is a species that is relatively distantly related to Argus compared to Cygnus. So the idea here was to test for the effect on the genetic distance of the reference species on the quality or accuracy of the assembly. Now, if any of these 3 strategies and tools were successful in assembling and circularizing the mitochondrial genome of the Caribbean spiny lobster, then we predicted the presence and we expected to observe in the assembly graph of each one of these, produced by each one of these tools, a circular molecule that is approximately the same size than the short read, approximately 16,000 base pairs. So we visualize the assembly graph of fly, unicycler, and reballer using the software Bandage. We identify a circular molecule of the correct expected size, and then we blasted this sequence, this contig, against the NCBI non-redundant database to confirm identity of the circular chromosome. And if we were successful, we finally did a final extra polishing using Medaka. Let me explain here the idea here. We know that Medaka has been established or has been developed for polishing contigs or assemblies when base calling actually is conducted with Guppy. But this was an experimental part of the protocol here. We wanted to know if Medaka helps in increasing accuracy when the base color is albacore. And the model that we use within Medaka was the most similar one to the version, the albacore version that we use for base calling. And the news is that, just let me give you a quick mention some good news. Medaka actually does improve accuracy of assemblies even when we use albacore for base calling. I will show that in short. So if we were successful in retrieving complete mitochondrial genomes using the strategies that I did mention, what we did was use different metrics for estimating accuracy. Completeness was one of them. The length of the mitochondrial genome, we expected, if it is a highly accurate, to be similar or identical to the gold standard mitochondrial genome that we generated in the study. Coverage was another metric. And finally, sequence identity. And specifically in this study, we use patristic distance as a proxy for sequence identity. We align using the software Muscle each one of the long read mitochondrial genomes against the short read gold standard mitochondrial genome, and then we calculated uncorrected p distance. And the idea here is that if p distance is 0, then the long read assembly is exactly identical to the short read gold standard assembly, and the larger this number, the more dissimilar is the long read assembly to the short read assembly. So in this first table, we have these metrics that I was mentioning you. The first thing that I want to highlight is that either using fly, unicycler, or reballer, all the strategies that we use did allow us to retrieve complete circular mitochondrial chromosomes of the species that we're studying. So that is good news. Remember, we're using a low coverage strategy here. And the retrieval of the chromosome is relatively quick. The length of the long read assemblies was very similar, regardless of the strategy we use, to the reference or gold standard mitochondrial DNA. Coverage was very similar, vary approximately between 30 and 40x, and p distance also was very similar. So in general, all the strategies that we use were successful in assembling a mitochondrial genome, and the accuracy looks like was very similar among the different assemblies. So to the question, can we assemble mitochondrial genomes quickly using long-read nanopore data sets? The answer is yes, we can. The next point is how accurate are these mitochondrial genomes, and I want to highlight that benchmarking of these long-read assemblies in mitochondrial genomes or even in bacterial genomes is not that common, it's not that frequently reported. So what I want to show you here in this graph is the number of sequence errors of these long-read mitochondrial assemblies depending of the tools that we use and the different pipelines, with or without medaka, with fly, different number of polishing cycles, with unicycler, different strategies, and with rebailer using different reference sequences. This is the number of sequences errors, and we have classified each error according to error type. More specifically, substitutions, one base pair insertions, short insertions or deletions too, and then we measure if errors also were one base pair to five base pairs homopolymer insertions or 1 to 5 base pairs homopolymer deletions. And one piece of information that you can retrieve quickly from this graph become obvious when you see the dominance of the green color here in this, regardless of the assembly and the strategy, the dark green color here is dominating in this graph. That means that the most common sequence error for the long read assemblies was one base pairs homopolymer deletion. And that's something that has been rarely reported, but has been actually other colleagues have reported that this to be one of the most common errors when assembling chloroplasts genomes with nanopore sequencing. The second most common error was a one base pair homopolymer insertion. That's the orange color that we see here. And the other type of errors were not that common. So again, very common error, homopolymer deletions or insertions on the one hand. And the other piece of information that you can extract here is obvious when you compare bars side to side, high, low, high, low, high, low, and that pattern repeats. And that's the effect of medaka, of extra polishing with medaka that I was mentioning. Even though we're not using the exact proper model for extra polishing, still medaka is improving accuracy when you do base calling with albacore on the one hand, and this improvement is particularly obvious for the fly um, assemblies. So we definitely recommend, if you're interested in the future, if assemble mitochondrial genomes using nanopore long reads, I will argue fly is the way to go. Turned out that we also tested Canu, and Canu, several strategies, we were not able to retrieve a circular mitochondrial chromosome with Canu. We don't recommend Canu, and we do know that Canu does have issues retrieving small circular molecules on the one hand. So in general, long-read genome accuracy using nanopore low coverage data sets is very high. However, this accuracy is not 100%. And in some way, the ultimate test of long-read assembly accuracy, we can have it if we annotate each one of these long-read genomes using Mitos and Mitos2, again, state-of-the-art bioinformatic tools available online for the annotation specifically of mitochondrial genomes. When we did that for the long-read assemblies, we obtained some very interesting results. This is, again, I already show you the annotation for the gold standard short read assembly, but for each one of the long read assemblies, all of the strategies we use, the number of errors was such that unfortunately the annotation with these tools wasn't totally reliable. In all of the genes but one or two, but one or two of the protein coding genes in each one of these long read assemblies did have at least one internal stop codon, and usually many, that did interrupt the open reading frame in each one of most of these protein coding genes, and that is indicated here by the black tone or color in these genes. So only a few genes actually were properly called by mitose. If you study, if you go and visualize, or if you manually do this annotation, you will see that the gene is there, but the open reading frame is really disrupted, and that is producing that the bioinformatic tool is actually not recognizing the presence, the position of that gene in the mitochondrial genome. However, it is there. So, although highly accurate, the errors contained in the long-read assembled mitochondrial genomes preclude, in general, generating a reliable annotation with mitos and mitos2, and that has a very important implication. I will argue that at this point in time, or let's argue during the year 2018, using the bioinformatic tools in this study, the quality of the genomes assembled using long read, though it's very high, it's accurate, is not that accurate. So, for instance, to proceed to study selective pressures in protein coding genes. Unfortunately, it is not there. I do realize that nanopore sequencing technology initial sequence error is improving by the month or by the year. So the hope is that maybe in 2 or 5 years, the quality of the genomes, mitochondrial genomes that we can generate, we can retrieve using nanopore long read will be high enough to proceed to do selective pressure analysis with the data sets and with the sequences that we generate using long reads. So last but not least, irrespective of the problems with accuracy on these mitochondrial genomes, the next question that I was interested in answering was if these long read assemblies that can be retrieved quickly and hopefully relatively cheap can be used for mitophylogenomics and for barcoding research. And then again, these type of studies are really important for understanding connectivity, for instance, in the Caribbean spiny lobster, or for detecting mislabeling on the other hand, and that will improve, that will help us managing the fishery more efficiently and towards the goal of sustainability again in the future. So this is a first phylogenetic livelihood analysis that we perform in the lab using a total of 42 terminals. This analysis rely only on protein coding genes, mitochondrial protein genes. We use a total of 11,187 nucleotide characters. Almost half, a little more than half of them were parsimony informatives. We use IQ3 for phylogenetic inference. And in this phylogenetic tree, we can see that all the long read assemblies, together with the gold standard, cluster into a single phylogenetic clade, fully supported by bootstrap analysis on the one hand, and this clade is sister to another clade comprised of Panulirus japonicus and Cygnus. And that actually, on the one hand, tell us that first, that long read assemblies, although they have problems about accuracy, enough to prevent selective pressure analysis, they do have high quality phylogenetic informativeness that will allow us, again, to conduct this type of analysis with this type of data in the future. The other important message that I want to comment here is that the bootstrap values didn't decrease considerably towards of the root of this phylogenetic tree. That is indicating that phylo or mitophylogenomic data or protein coding genes of mitochondrial genomes exclusively maybe can be used to reveal phylogenetic relationship among closely or distantly related species in lobsters. And the point is that definitely we know that the mitochondrial chromosome is just a single marker. We are interested definitely in using nuclear genomes, but my point here is that mitochondrial genomes can provide you with a lot of reliable information to reveal uh, phylogenetic relationships among groups of closely or not so closely related species. In this case, in the same genus, family, or even infraorder in the case of this crustacean clade. A second analysis that I want to show you is again a phylogenetic analysis. In this case, what we did is we retrieved from gene bank the totality of the sequences belonging to the cytochrome oxidase 1 gene available in gene bank and towards the during the end of the year 2002. We add our COX-1 sequences for a retrieve from the long read assemblies, from the gold standard to conduct this phylogenetic analysis. And what you can see here, as indicated by the black arrow tip here, is that again, all these CO1 fragments or COX-1 fragments retrieved from long read assemblies plus the gold standard fragment clustered together into a single well-supported clade that was contained within another clade comprised of exclusively specimens of Panulirus argus, and this clade was clearly differentiated from others comprised of sequences belonging to other species closely or not so closely related um, uh, lobster species. So again, the question is, can these fragments retrieved from a barcode or retrieved from long read assemblies can be used for barcoding analysis, although we know they're not completely accurate? The answer is yes, we can use them. I want to highlight that the great majority, 99% or more of the sequences available in gene bank of this COX-1 gene actually are sequence using PCR and Sanger sequences. So these are relatively high quality sequences compared to these long read sequences. So a combination of them for this analysis can be permitted in some way. So a main point here is that of these last two analyses, although not completely accurate, long-read mitochondrial genomes can reliably identify, in our case, the sequence specimen as belonging to the species that we're studying, Panulirus argus, and also can differentiate our specimen from other closely and even other distantly related species in the same genus, family, and infraorder. So again, the answer to the question, can we use long-read assembled mitochondrial genomes for barcoding and mitophylogenomics? The answer is yes. Before I finish, let me give you three main messages. First, this study, I hope, turned to be a proof of concept for the future implementation of in situ surveillance protocols using the Minion to detect mislabeling in the Caribbean spiny lobster across its supply chain. The idea again is having markers that maybe they are not perfect, but they have high accuracy, and that they are cheap and that they can be retrieved quickly to surveil mislabeling, so to improve the management of this fishery. The idea also with these studies to hopefully decrease the cost for exploring metapopulation connectivity in the Caribbean spiny lobster. We have already developed a panel of SNPs based on 2B rat sequencing. That's a study that we published a couple of years ago. And the idea is to use mitochondrial genomes together with the SNPs to understand connectivity among populations in this species, migration, demographic history, and of course, hopefully adaptation, mostly relying on SNPs. And last but not least, actually, the idea with this study is to hopefully use This specifically not PacBio, that may be nanopore sequencing technology. So to transfer it to colleagues in moderate or low-income countries, the Caribbean spiny lobster is present in all the Caribbean coast of Central America, the Caribbean, and even North South America. There's not too many resources in many of these countries, and the idea is my lab going and providing this resource, and hopefully together with colleagues in Caribbean and Central American countries to understand the connectivity of this very important species. If you're interested in additional details about this study, please, you're welcome to download this paper that was published at the end of the year 2020 on BMC Genomics. It's open source, of course, and available for you for free in the internet. And also I want to mention that as we speak, we are assembling the nuclear genome of the Caribbean spiny lobster. And as we speak, we are finalizing sampling of this species with colleagues in other countries in the Caribbean and Latin America, so to explore or to understand metapopulation connectivity in this species, local adaptation, and again, with the goal of improving maybe the fishery of this really important species. If you have any question, please visit my website at Clemson University or send me an email. Thank you.